Okay, thank you so much, Carlo, for the invitation. And I'm happy to present this for the first time since the bioRxiv came out. So it's the result of my PhD work in the Trade-Line lab. And yeah, the topic is decoding morphogen patterning of human neural organoids with a multiplex single-cell transcriptomics screen. And I want to start first by introducing you to the team that I've had the pleasure to work with. Barbara and Gray were my PhD supervisors. Ryoko, Makiko, and Gosha have helped me with experiments. Akanksha has helped me a lot also with the brain part of this experiment design and so on. And Jisong, Jasper, and Jonas were my computational helpers. Jasper was running Scenic for me, and Jisong provided a lot of advice for computational matters, and Jonas helped with mappings to human tissue. And I'm also going to present a collaboration with the Kirkeby lab. Pedro, Charlotte, and Gaurav also contributed generating the data that I'm going to show you. So now let's get into the topic with this beautiful animation of a developing human embryo, just to highlight how fascinating it is that a single fertilized cell can become such a complex organism. And the brain is one of the most complex organs in the body and what we believe that makes us human. So we want to study how the brain acquires those unique features, but we still have very limited understanding of the process because we don't have primary tissue and we have ethical implications on the way. But what we do know is that the neural tube is the precursor of the entire CNS of the central nervous system. So as early as 4 gestational weeks in humans, the neural tube gets segmented into the forebrain, midbrain, hindbrain, and spinal cord, which will subsequently divide and result in more compartments. And by 24 gestational weeks, they will give rise to this perplexing variety of specialized structures that you can see in this schematic. And how does this happen? This great regional diversity is achieved thanks to morphogens. And morphogens are molecules that are secreted from a specific set of cells during development, and they diffuse through the tissue, and they form concentration gradients that specify the fate of each cell along the gradient. But only the interplay of all of these can give rise to the perplexing variety of regions in the human nervous system. So how do we learn about morphogens and what they do? So far, we were relying on mouse models, but for obvious reasons, as you can see, the difference in size, we needed something better. Also, mice have divergent cytoarchitecture and gene expression profiles. So 2D neuronal differentiation platforms came up on the way as a possible alternative, but they are very basic in arrangement. They're unstable, only last short term in culture, and they don't really recapitulate human development on a dish. So here came organoids to the rescue with improved tissue architecture. They have more stability. They can be maturated in vitro for a long time. And they importantly recapitulate key features of human brain development, as many papers have shown so far. And another important thing is that they are a very tractable method that allows for manipulable genetics and culture conditions. So it's a perfect setting to study morphogen effects on a neuroepithelial sheet. And the way neural organoids are grown, as most of you know, is starting from human pluripotent stem cells that get aggregated into embryoid bodies, and then they are exposed to neural induction factors to specify neural lineage, and then we further promote neuronal differentiation. If we follow this kind of protocol and we let the organoid be, we would obtain an unguided neural organoid that could generate many different neural regions depending on various factors. For example, the human pluripotent stem cell line of choice. But if we add some patterning molecules or morphogens, we can direct their regionalization, and we can generate region-specific organoids like the ones that you can see here for specific parts of the CNS. And these are just 3 examples for the main subdivisions of the brain. But actually, so far, many groups have created their own protocols to generate regionalized organoids. And it seems like there are many ways to generate the same thing, or at least that's what we think. But each of these existing protocols use really divergent culture settings. So for example, the neural induction methods are different. You can see, you cannot see my cursor, okay. On the panel on the left, you can see neural induction methods employed by all of these summarized organoid protocols. They use activation and inhibition of different pathways to achieve neural induction. They also use different patterning molecules sometimes to achieve the same regions, as you can see on the panel on the right. And then there are other factors that I'm not going to talk about today, but they also play an important role in organoid development, like static culture versus shaking culture, extracellular matrix. The presence of extracellular matrix might have a huge impact. Akansha will talk about this later, and also maybe the addition of maturation factors. So, as I say, these effects can go in many directions: regional patterning, cell type diversity, maturation, tissue architecture, and also in addition, so far the development of new organoid protocols was a very inefficient process. The screening was just based on bulk measurements of a few marker genes and just with a very low throughput. So we thought we needed to build an integrated reference of human neural organoid patterning to assess the role of these morphogens and also enable the development of more and more specific and reliable regionalized brain organoid protocols. So we decided to focus on these main morphogens, the main characters of the regionalization process: sonic hedgehog, the ventralizing agent. We use cyclopamine to inhibit it, recombinant human sonic hedgehog to activate it, FGF8, which is mostly secreted from the anterior neural ridge at the very anterior part of the neural tube and in the midbrain-hindbrain boundary between the midbrain and the hindbrain. Retinic acid, which plays an important role in the segmentation of the hindbrain. And we also used agonizers of the Wnt pathway and antagonizers here and XAV, namely. And we also modulated the BMP pathway. So we touched on BMP4 and BMP7. And this is the way our assay looked. So it's a plate-based assay where we would culture organoids for 21 days. We would have 14 days of neural induction and then another week of neural differentiation. And we would grow, in general, 8 organoids per column, but it varies per condition. And after 21 days, we would take each of these conditions and we would dissociate each of them and then hash them using the method published already in 2017 to multiplex everything together, run it in on a 10x, and then obtain single cell profiles of each of these conditions from pooled organoids, importantly, I should say. So it's 4 to 20 organoids pooled. And what we wanted to look at was basically a very systematic view of things. So we looked at timing. We designed different timings all the time, the same for all of the morphogens, concentration steps. Once we determined the timing, the patterning window that we were interested in, we also tested the concentrations, and we also tested the interactions of morphogens as they would happen in the developing neural tube. And then also we touched on different things. Some of them I'm not going to talk about today. So neural induction modes and HPSC line variability. This is a bonus that's in the preprint, and maybe if there's time I can talk about later. But I'm going to talk a bit about patterning modes. So microfluidics, so gradient-based patterning versus organoids or stepwise patterning. And here I can show you the outcome of all the conditions that we tested. So this is a UMAP projection that has all the cell types that we obtained. Since this was an untargeted approach, we obtained both neural cells and other cells out of the neural lineage, and also neural crest. So we have the CNS progenitors at the top and CNS neurons. You can see the expression profiles of each of these marker genes on the right. And if we zoom into the CNS neurons, we can also see the different subtypes corresponding to the progenitors of the central nervous system that are in that pool. So we know that they are maturing. And 21 days is like a good time point to look at progenitors and also at neurons. We were mostly interested in patterning of progenitors. So here, I just want to show you like a broad overview of the research we put together. So you can see variations in cell type composition in different timings, in response to different timings, response to concentration, variations, stepwise variations, and combinations of morphogens with the corresponding controls that's indicated by like the bracket. And I will delve deeper into a particular example later. But also, so we have this cell type-based analysis that tells us a lot of what's going on. But to back it up, we know that biology is noisy, and we wanted to have also a regulon-based analysis. So we built this network of associations between regulons and patterning molecules using CINIC. And this allows us to see what's going on below the cell type specification level. So in this network that you can see, it's very complex. I know it's like a lot of information, but I can tell you, so the edges, the lines that are connecting each of the molecules with the corresponding regulon, the color, the intensity represents the correlation, the strength of the correlation, so the weight. And then the size of the nodes represent the regulon size. So there are some regulons that are bigger than others. And the color represents the cluster that they are most enriched in. So for example, for retinoic acid, you can see many regulons that are associated to spinal cord or posterior floor plate cells. And it's cool to see that each of them have a specific set of regulons that they regulate, very redundantly speaking. But also some of these regulons are controlled by several of these molecules. And this is important to take into account. And to understand this a little bit more, we also calculated correlations of each of these associations to timing and concentrations. So for example, you can see how each of these molecules is varying with time and with concentration changes. On the right, you can see that sonic hedgehog has some branches that are very yellow, meaning it's highly correlated to concentration. So the higher concentrations that you give of sonic, the more those regulons get activated. And I will give an example, a clear example later, because there's too much information and there's no time to go through all of them. So I'm going to start with retinoic acid. This was the most robust morphogens that we had. And for the timing experiments, we could clearly see patterning windows, first for non-neural ectoderm in early days. And you can see the top two bars. We would enrich for non-neural ectoderm if we gave it early. And if we gave it late, we would enrich for hindbrain progenitors very slightly, and then for retinal progenitors. And the dose that we use here was like a medium dose, like you can see below the days panel. It's concentration 2/3. So that's a medium concentration. And if we take the later time points to pattern, so the neural time points to pattern, and we do the concentration steps, we can see something very interesting. Now we get posterior floor plate. If we increase the concentrations, we also get hindbrain progenitors. The higher the retinoic acid, the more hindbrain progenitors that we get. And retinal progenitors are depleted at higher concentrations. And we can see that the lower ones that we were using earlier in the timing experiments are appearing only, like retinal progenitors are only enriched with lower concentrations, which matches the timing experiment. And we can see all of this summarized in this panel, which is based on the regulon analysis. So you can see this map contains all the regulons that are associated to retinoic acid. And on the y-axis, we have the correlation coefficient to concentrations. And on the x-axis, we have the correlation coefficient to timing. So if one regulon is very high and to the right, it means it gets activated with higher retinoic acid and with late retinoic acid exposure. So it goes a bit like the more correlation to concentration, the more it gets activated with more morphogen, and the more correlation to timing, the more it gets activated with later time points. And you can see that here retinoic acid has its regulons all over the map, meaning that both time and concentrations matter a lot. And I can put this into perspective by comparing with BMP4, for example. In this case, all the regulons that are associated to BMP4 are scattered only across the x-axis. So this means that it's only correlated to timing, the activation of these regulons. And concentration didn't really play a role in the effects that BMP4 would cause. Okay, now I'm going to go to the combination experiments. So because in vivo, we only get the regions by the interplay of all the morphogens, we needed to test this in a very similar scenario. So we designed this coordinate system to test interactions of morphogens that would normally interact in a neural tube. So we would antagonize Wnt with XAV in the anterior part to create forebrain. We would use KIR to activate Wnt in the hindbrain, and then retinoic acid for like hindbrain segments as well. And we would also use sonic and cyclopamine A to modulate the dorsal ventral axis. And we saw something interesting if you look at the organoids that I'm highlighting here. So there was an interesting interaction between retinoic acid and sonic hedgehog. So here, the organoids that get sonic hedgehog alone would not produce any posterior floor plate nor hindbrain progenitors. The organoids that get retinoic acid would get more posterior floor plate than hindbrain progenitors. And then if we add sonic hedgehog on top, it would boost the production of posterior floor plate. Then we added FGF8 on that side of the grid. So on the segment that we expected to create hindbrain, because FGF8 is secreted from the midbrain-hindbrain boundary. And if we look at those organoids, now we see that the effect is reversed. So now if you put FGF together with retinoic acid, you get more hindbrain progenitors. But if you add sonic hedgehog again, you get more posterior floor plate. So you can use the ratio of each of these morphogens to get your desired outcome. And these combinations that I've talked about are, of course, stepwise changes in concentration. So we are crossing X concentration of one morphogen with Y concentration of another. But in vivo, what happens is that we have gradients. So we focus on the forebrain as the system that we wanted to study. And in the forebrain, what happens is that Wnt and BMPs at the dorsal side counteract that gradient from the bottom, from the ventral side of sonic hedgehog, and SFRPs in the middle counteract the Wnt from the top. And this results in different patterns at the anterior and the posterior part of the forebrain. So we would get the cortex at the top, and then ganglionic eminences in the middle and preoptic area in the anterior part, and in the posterior forebrain, we would get the thalamus at the top and hypothalamus at the ventral side. And how do we recapitulate all of these in vitro and see what we can achieve with organoids as well? Well, we turned into this microfluidic system from the Kirkeby lab, as I said earlier. This is a published method, and it's a chamber where you can basically mix two different medias and obtain a gradient of your desired morphogen. And in our case, we used a dorsalizing media without any factors and a ventralizing media with sonic hedgehog agonists. And we cultured this tissue, which is like a neuroepithelial sheet, in the same way as we cultured the organoids before, as you can see in the bottom, for 21 days. And we compare all these with these continuous concentration steps of organoids. with the aim to see whether the sonic Hedgehog gradient was actually necessary for complete dorsoventral patterning. And we assessed all this also with the hashing method that I presented in the beginning. And when we put everything together, all the cell types that resulted, the integration was really remarkably good between the organoids and the MR system. As you can see here in the first UMAP, the only island that seems to be more the organoids is the bottom, which I will show you later what it is. And since we used hashing of the different segments of the MR tissue and also the different concentration steps of the organoids, we can also trace back what are the dorsal-most regions and what are the ventral-most regions, which you can see in the UMAP on the right. So the blue ones are the most dorsal and the orange ones are the most ventral. And now if we look at the cell types that we generated, we have some cortical, pallial, subpallial boundary, and like ganglionic, lateral ganglionic eminence progenitors present. We also have some MG progenitors, and we have preoptic area and hypothalamic progenitors. And we also have FOXA2 cells. Both systems, both the continuous gradient and the discontinuous one in organoids could generate these cell types, but the continuous morphogen gradient, namely the MR, was much more dorsalized than the organoids. And these we can also see using this analysis. So we calculated a four-brain axis score where we could rank the cells according to their expression of markers for the dorsal-most and ventral-most regions. And here you can see, for example, the MR, most of the cells would correspond to more dorsal scores, and the organoids would correspond, the cells of the organoids would correspond to more ventral scores. So MR has more cortical, pallial, subpallial boundary LG progenitors, and organoids have more MG progenitors and hypothalamic progenitors, preoptic area. So it's all shifted, and we think this is because of the PDMS absorption of the system. But in general, we were happy to see that the organoids can actually recapitulate the entire continuum. So there was no discontinuity in the cell types that we could obtain. It's just shifted more ventrally, which we can account for by giving less sonic. And on the left, you can just see the images of the organoids that have NKX 2.1 lighting up on the A condition, the dorsal most, whereas the MR tissue on the bottom has more NKX 2.1 at the ventralized region, so CDE segments. Okay, so yeah, with this, I'm gonna wrap up. So to summarize, we created an integrated reference for human neural organoid patterning that hopefully it's useful for many people. We studied systematically many variables that can affect the outcomes of morphogen patterning. And we used a multiplex platform that allowed us to look at the cell type and regulon level. So it's a bit of a more robust method to survey for new protocols. And what I presented to you hopefully made it clear that we have now some competence windows delineated for the main patterning factors that we were testing. We also saw that morphogen dosage can regulate cell fate decisions, and you can use it to tune your cell type of choice. And also we observed some interactions, interesting interactions between morphogens. I only talked about the one with retinoic acid and FGF and sonic Hedgehog, but we also saw some with sonic Hedgehog and the Wnt pathway. And then the last thing that I presented to you is that organoids can also recapitulate the continuum of cell types in the dorsal-ventral axis of the forebrain. The last things that I didn't have time to talk about are cell line variability and neural induction methods, which can both impact, as you can imagine, probably, they can impact the outcome of your morphogen treatment. And with this, I want to thank you. Thanks to Carlo for the invitation. Thanks to everyone who's listening. I want to thank the team that I presented before, the entire Choitlan and Camp Lab, and the people at the MPI that I worked with at the beginning of my PhD. Also, Nadia from the Trailline Lab will now pick up with some revision experiments, so she will join the main team. And also the Kirkby Lab, who were extremely helpful with the MR experiments, and it was great to collaborate with them. That's it.
Great, thank you so much. Any questions? I might prompt you to give us maybe a touch more information on when you mentioned there at the end different lines. I know that there's probably a lot to talk about there, but could you give us the short version of maybe
Sure.
what you saw there? Looks like you tried 3 different lines in this system.
I'm happy you asked because I didn't have time to include it, but I think it's very interesting. So you can see my screen, right?
Yeah.
So here I have this panel, and I want you to guess what are the columns and what are the rows? Is it the HPSC line or is it the morphogen treatment? If you have a guess, I'm gonna now see if you did it right. So rows are cell lines,
Wow.
and columns are treatments. So we cannot override the morphology that one particular cell line tends to have. Probably the morphology comes from the different SNPs that line might have, and epigenetic features specific for that line that respond to factors. So
that's really incredible.
It's all probably about factors, but it's very striking that HES3, for example, is all round, and we see no change with FGF low or high versus control. And then WTC and H9 also have very remarkably different morphologies. And then we also, sorry, you want to hear more, or should I stop?
Yeah, actually, another question before you take another step. In this particular experiment, I know you mentioned that you varied the timing of morphogens. Relative to the onset of neural induction in this particular experiment, when did you hit them with the listed morphogen there, the FGF8?
This one, we gave it late, because for FGF, we tested both early time points and late time points with an extended time window for concentration steps. This has like many layers of complexity. I hope it's not getting confusing. But early on, FGF usually promoted telencephalic identity, but more ventralization than anteriorization. So Sonic was more powerful in making anterior tissue than FGF, which was surprising to us, because usually FGF is secreted from the anterior most part, and you would expect that it makes telencephalon. But yeah, here, this specific experiment is when we gave FGF later on, because we would expect that this one would give us effects on the midbrain hindbrain boundary. So that segmentation occurs later, so we gave FGF later, just to act on that specific patterning event. But you need other events happening before, so you cannot ensure that an organoid that's only seeing FGF is gonna create a midbrain hindbrain boundary. So yeah, this one, this particular time point was day 9 to 20. So that would be spanning neural induction and neural differentiation stages. So it's late in the neural induction time, but it is also in that slot.
Thanks.
Yeah. And
so
yeah,
I think in the interest of time, we can move on to Akanksha's talk, but thanks so much. And I'm anxious to hear more about these line effects as you see more. Okay. Oh, we do have one online question. Let's hit that before we go. So Dashway Online asks, are all cell lines cultured in the same PCS media systems? And have you explored patterning effects across different pluripotent stem cell culture conditions in the MR versus E8?
Yeah.
And teaser versus E8.
So we homogenized, so we actually adapted all our cell lines to IPS brew, because we were also using IPS brew for the H9 that we were using in the Mister cultures. So all these cell lines were cultured in IPS brew, which was actually quite a nice pluripotent stem cell media, I would say, better than all the others. It was very robust for all of them. We didn't test differences between different stem cell media. And what was the other question? Yeah, like we didn't really explore on the PSC side. Like there were enough things to explore.
Enough variables moving.
But yeah, I'm sure there are differences.
Thanks so much, Fatima.
No worries.
Excuse me, Carlo. Is that possible to just ask a very quick question?
Sure.
Just a very general question, considering you have so many different variables here, from the morphogen to the different lines. I just wonder, and then you have demultiplexing, single cell, etc., etc. I just wonder which of these you find it the most challenging one. If you want to repeat the experiment, you're just gonna, for example, drop the two lines, you're gonna go with one, or no, what was the most challenging part of the entire experiment, considering all these variables?
So I have to say this was not a single experiment, but it was many batches that were grown separately, and we have replicates for some of them. For some of them, we don't. So we're now working on the revision experiments, and we will do one big experiment where we will look at two different batches, but in the same, not this time we're going to use parse for single cell readouts. So we will pull all that together, but it will be two different batches. So in different incubators with different media, different starting pool of stem cells. And yeah, basically like the usual variations that you would see batch to batch. And we're reducing to lowest and highest concentrations, because we did here the stepwise variations so that we can use this for modeling in the future. I think it would be super helpful to have the data, because it's like a continuous readout. And you can like trace a line sort of and see where your line will go if you give more or less of the morphogen. But yeah, the experiments that we're doing then just go to the highest and the lowest concentration. And then we're also completing on Kier timing experiments. And
you're still going to use the 3 lines?
Yeah, so we use 3 lines. In the beginning, we were using HES3 only, but HES3 is a reporter for NKX 2.1, so we could assess a little bit quickly what was happening there. And now we will use 4 lines actually, and we're accounting for ESC versus induced pluripotent stem cell. But I think this requires a completely different study that just goes on that, and it has to be really thorough, because you have to look at each of the cell lines epigenomic profiles to know what's accessible, what's not. And here we don't have any of that data, so I think it really needs to go much more in depth. I think that would be most likely. Thank you.
Thank you very much.
Thanks so much. So I think we can maybe do more questions at the end, so make sure Akanksha has time to do her presentation.